Good evening, and welcome to review for our first test. Biochemistry, macromolecules, water, and some basic chemistry. So let's start off with our friends, the carbs. Carbs, we know, are going to have a basic structure. They're going to have carbon, they're going to have hydrogen, and they're going to have oxygen. They will be in a, let's say, X 2x to x ratio. So for example, in something like glucose, glucose will be C6H12O6 with our hydrogen and oxygen in a 2-to-1 ratio. Carbohydrates are going to probably be written in a ring structure. So if you look at the glucose, you look at the galactose, and you look at the fructose, you will see there that they are in a ring. And in some cases it has 5 sides, in some cases it has 6, but either way there will be 6 carbons. These things are called isomers. Isomers are simply the same exact chemical formula of C6H12O6, but a different orientation. If you move down and you take a look at how you build or how you go from a monosaccharide or a monomer to a polysaccharide or a polymer, in this case we have glucose and fructose coming together to make sucrose, your common sugar you may have in your kitchen. In order to do that, there's not enough bonds, so you have to remove something. So if you look here, you will see that the H2O disappears, freeing up bonds, making what's called a glycosidic linkage. That is found right here. Since the water left, this is called dehydration synthesis. We will see this in the creation of all macromolecules, removing the water and adding 2 monomers together, or in the case of lipids, 2 sort of kind of molecules to make one larger one. We are going to talk about starch, which is the storage in plants, glycogen, storage in animals, and cellulose, which is structural in the cell wall of plants. Plants will use starch as a storage for energy, which we just said, and things like potatoes and carrots, and we can break that down. Glycogen is where we're going to store our energy, and we are going to store it in the liver or in muscles. So those are places that we may see our polysaccharide glycogen, or, well, we'll see the glycogen there, and plants will store it in the roots. We have enzymes to break down things like starch and glycogen. And we have the enzyme for these things called alpha linkages. And in alpha linkage, let's say that this is starch. And we really don't know how many monomers there are. And it says here there's 300 to 600 right there. So this is a large molecule. And in order to break it, we do have the enzyme. And that enzyme will act right here with water, because it's going to be hydrolysis to break up this bond. And that's how we can free up the monomers in a polymer of starch. In the event that you are an animal that eats primarily grass, there's a lot of cellulose found in grass, in the cell walls, which we talked about. And some animals like cows or termites, their diet is very heavy in cellulose, whether it is wood or it is grass. If you look here at cellulose, you can see that the OH group here alternates. So there's what is known as a beta linkage. And we don't have an enzyme for that. So in order to help animals take care of this, they have microorganisms in their gut. They can be anything from bacteria to protists. And they have a mutualistic relationship with these larger animals. So although a cow may chew and re-chew up the grass, that will do nothing without the help of microorganisms. Other structural carbs you may see are things like chitin, and that's going to be kind of like a modified carb, and that will be found in the cell wall of some various fungi, as well as some invertebrates like bugs and crabs. So here is the isomer again. We said that isomers have the same molecular formula. In this case, this is a 3-carbon sugar. But because the orientation is different, these are called isomers. Okay, let's see, moving along. Now we're moving our story over to proteins. Now proteins are also going to have carbon, and they're going to have hydrogen, and they're going to have oxygen. But proteins now, more than carbs, are going to have nitrogen, and some, as in 2 of the amino acids, could contain sulfur. So in any protein, the monomer is an amino acid. Amino acids will show this structure right here. And with that, you will see a central carbon. Off of that central carbon will always be a hydrogen, and once again, they may draw it above or below. That does not matter. That is your R group, or your functional group. There'll be a carboxyl, or the C terminus. The amino group, otherwise known as the N terminus. And sometimes they will put that extra charged hydrogen there, but as a general rule, that will not be there. But sometimes they will draw it in there. The H, once again, or the hydrogen, is always bonded to the central carbon, and the R group will come off of that central carbon as well. This is what will give each of the 20 amino acids a very specific characteristic, and that could include polarity, that could be hydrophobic, or hydrophilic, and each of those characteristics for those R groups will determine how and where the protein folds and if it will function. In order to build a protein, we use dehydration synthesis once again. So here you can see there are 2 amino acids. They're not named because they each have just a wildcard R group. But in order to put them together, you just can't add an extra bond. It doesn't work that way. So you need to remove an H2O, dehydration synthesis, and you're going to form what is called a peptide bond. And the peptide bond will always be NCC, peptide bond. NCC. If there was another amino acid, there would be a peptide bond right there. It's not. This will be called a dipeptide because it is composed up of 2 amino acids. This is not a protein yet. We are just building the structure. Moving along, proteins are going to come in many different orientations, and some will have very different functions. They could be used to transport in the form of hemoglobin. They could be hormonal and insulin. They can be enzymes. That's going to be your major giveaway. They could be structural. They could be help in cell communication. And in cell communication, they could be receptors. So proteins are a wild card, and they come in all different shapes and structures. All proteins, however, will start with what is known as the primary structure. That is simply the sequence of amino acids, which is determined by the DNA itself, with some help from RNA, taking the message out to the ribosomes. So if your ribosomes get damaged through, I don't know, radiation or chemicals or any of that, no ribosomes equal no proteins. So that's extremely important. We will see more in the next chapter on that. Secondary structure, things are getting a little bit more interesting. Here, we are looking at the backbone chain, and what I mean by the backbone chain is the NCC part of the chain, not the R group. Here, you're going to form hydrogen bonds, and you will form with those hydrogen bonds either alpha helices or beta pleated sheets. So if we draw that real quick, well, we don't really have too much room, but an alpha helix will look something like that, and the beta pleated sheet will look something like that. And it is the hydrogen bonds that are stabilizing it in through this region. The R groups are no R groups are not involved. Tertiary structure, now you're on the 3rd level. Here things are getting a little bit more interesting. Here we're looking at a 3D structure. And here it is all about the R groups. There could be disulfide bridges. Your hydrophobic amino acids are going to stay in the middle of the protein. They're going to stay away from where water may be. Your hydrophilic amino acids are going to be on the edge where they will interact with water. Any mutation that happened due to DNA issues, whether they were inherited or picked up through nearness to radiation, will possibly cause the DNA to be wrong, which will cause the RNA to be wrong, which will cause the protein to be wrong. In that case, switching out amino acids with different R groups could cause different folding patterns. And we know that the shape determines the function. So if they're not shaped correctly because of the correct order of amino acids, the protein may not function. Quaternary structure here will be more than one protein chain, aka 2 or more. So a good example would be something like hemoglobin, where you will have, 1, 2, 3, and draw some black in there, 4, and you'll have 4 chains all working together and interacting, and any one mistake could cause a major problem. So in the case of sickle cell anemia, which is a genetic disorder, if one hemoglobin is not functioning correctly, it is the interaction between hemoglobin molecules, so between them, that's the problem. It's not within. Within the molecule, it's a little messed up. It's one amino acid mistake. But because of that, the interactions between them is what's going to cause the sickle cell anemia or the sickling shape of the red blood cells. We will also learn a lot more later, but that in the event that the temperature is not correct, too hot, too cold, or the pH is not correct, too high, too low, you will get what is called denaturing of the protein. So let's say that it looked like that, and now it unfolds, and if it unfolds, that's going to be a problem. So anytime it unfolds, it stops functioning. Not breaking it up with water or hydrolysis. That's way more than this. That's going to come with different enzymes and all kinds of things. Here we're just talking about messing up the structure the way it looks, denaturing, irreversible. Now we move our story to lipids. Well, lipids now have, just kidding. Yes, I'm sorry. Lipids are going to have carbon, hydrogen, and oxygen as their general rule. We're going to put this phosphorus here, gets a specific star right there, specific. That's only in a modified lipid called a phospholipid. But whenever I talk about lipids in general, if I do not mention phospholipid, we are assuming regular lipid, carbon, hydrogen, and oxygen only. And the C's and the H's are way higher than the ratio of oxygens. So that's how you know it is a lipid. It is not a 1 to 2 to 1. Lipids include waxes, oils, fats, phospholipids, once again, asterisks, we'll see more about those in a second, and things like cholesterol. These things are non-polar, non-charged, and very hydrophobic. They do not want to know anything about mixing with water. Not at all. Things like cholesterols could be found in the cell membrane. Now, although they are not good for us in excess, we do need them for the cell membrane, and they are part of some hormones. So although eating too many is bad, we do need cholesterol for cell membrane maintenance and hormones. Lipids are going to have a ton of energy, way more than any other organic molecule. That's why animals like whales have such large amounts of blubber that will allow them to survive long periods without eating. Will also help in buoyancy, and that means to float, and keep them warm, or insulation. We will look at triglycerides here. Tri means 3, but it's actually being bound to a glycerol molecule, so there's technically 4 things involved here. 1 glycerol, 3 fatty acids. This is not a macromolecule of monomers like we learned about. It's macromolecule, but these are not monomers. These are individual molecules that are being added. Nonetheless, you're adding 3 things. 1, 2, 3. you're adding it to the glycerol, which is going to be written upside down, long ways like that. Well, you're adding 3 things. That means we're going to lose 3 waters. If you add 2 things, we lose 2 waters, and so on. So this here is going to be a saturated fat, and you can see that because there are 0 carbon-carbon double bonds. So this would be a saturated fat. And we know that saturated fats are solids at room temperature. Solid at room temperature. And we know that because here these fatty acid chains are going to pack nice and tight, and they once again will form a solid at room temperature. If you heat them up, they'll melt, but at room temperature, they're solid. And that's the glycerol with the 3 fatty acids. Once again, these are saturated. Coming over here, we see that there's no double bonds. We got that. If you are looking at a saturated fat, it's going to have this formula. C to the N, let's say that's 10. H to the 2N, that's going to be 20. That means every time you have a carbon, let's say for example like that, it's going to have 2 hydrogens branched off of it. And notice that ratio is way higher than that little 2. So it's a way higher carbon to hydrogen ratio than oxygen. That's saturated. That rule will not work in an unsaturated fat because unsaturated fats have 1 or more double bonds. And the 1 or more double bonds reduces the amount of hydrogens that can fit. These guys will be liquids at room temperature, like oils. In this case, you can see this double bond right there. Causes a slight kink in the fatty acid chain, which will cause it to be a liquid at room temperature. Now, various unsaturated fats may have more than 1 carbon-carbon double bond, and if they do, that will affect taste, that will affect melting point, that will affect a lot of things. And just to remember there, if you bond 3 fatty acids to 1 glycerol, that's a total of 3 waters. Now let's just mess around here, and let's say that we have a glycerol that is, I don't know, let's call it C3H8. Let's see, let's see how many oxygens we're going to have on that. We're going to have 1, 2, 3. Oh, 3. So let's say that this is our glycerol molecule. And again, I have to give this to you. I'm not gonna make you memorize that. And let's say that I have 3 fatty acid chains. 3 fatty acids. And let's say that, let's make that up and say it's C15H30. So we know that it's going to be a saturated fat. And let's say, what do we want to put at the end there? Let's put, I don't know, O2. Whatever, it doesn't matter. Now I got 3 of those, so I have to remember that my total is going to be C45, because I tripled that, H90, because I tripled that, O6, because I tripled that, plus my original glycerol, which is C3H8O3, for a total of C48H98O9. But I can't forget that I have to subtract 3 waters. And I have to subtract 3 waters because I put 3 fatty acid chains on there. So 3 waters is going to be H6O3. We're going to minus that. So my new formula, if I was adding everything up, would be C48H92 and O6. And that's how you add up the total molecular ingredients of a triglyceride if all of the numbers were given to you, which they will be. It's not fair to not give them to you. Okay, lipids continued. Phospholipids, now these guys get the asterisk, so they're lipids, but they're not in the true lipid family, which we talked about. Here, these guys are going to form the cell membrane. Now, cell membrane is extremely important because it's a lipid bilayer, which means it's 2 layers. And we'll draw it something like that, and something like this, something like this. This is the head, which is hydrophilic. These are the tails, which are hydrophobic. So because of that, they're called amphipathic, which means they have a polar end and a nonpolar end, or a hydrophobic end and a hydrophilic end. That becomes very important in the world of the cell membrane. Again, we'll talk about that a little while later. And when you look at this, they have a phosphate group right there, and there's your two fatty acids. So it sort of kind of looks like what I drew. If you look at it on a true molecular formula, there's the glycerol right there. There's your fatty acid chain 1, fatty acid chain 2, and there's your phosphate group. So that is your phospholipid. And that is going to once again go on to build a cell wall. So if you look at a cell wall, for example, like that one, no water here. How come no water? That's hydrophobic, so no water. But what if I had a really cool channel protein like, I don't know, something like this? Now, channel proteins are going to allow things to get through the cell membrane that normally wouldn't. So let's go ahead and draw the rest of my lipid bilayer there. And it looks something like that. There's my tails like that. Okay, so now if this is the situation, and I know that this blue thing is a protein, I know it could be made up of hydrophobic or hydrophilic R groups. So what you have to know is that in this region right here where I'm going to use green, anywhere I put green, that is where you're going to have hydrophilic R groups. And because they are potentially going to come in contact with water, especially if water is the molecule going through, which it might be, those have to be hydrophilic. Which means, let's go to, I don't know what color you guys want to use here. So we'll use red so you can see it. Now these amino acids in this specific spot only, they're going to be hydrophobic. And they're going to be phobic because they are in with the tails. And if you mix and match, the protein's not going to sit right. And if protein doesn't sit right, it's not going to work. So that is very, very, very important to understand why that works. Okay, so we got over there. Let's move our story along to nucleic acids. Now, nucleic acids have a player, and the player here is phosphorus. Now, phosphorus is always there. Always, always, always. Notice, no sulfur. Okay, no sulfur. So, as soon as you see sulfur, proteins. When you do not see sulfur, nucleic acids. If you see phosphorus and nitrogen, you're thinking nucleic acids, unless I go out of my way to say phospholipid, which I will do. Here, DNA and RNA, you have a phosphate group, so there's your P, obviously, right there. You have a sugar, and you have a base. Together, this thing is called a nucleotide. The whole entire thing is called a nucleotide. And you're going to put a bunch of nucleotides together, and that's going to build a nucleic acid. The carbons are numbered, but for this case, you're not going to have to worry too much about that. You have different kinds of bases. You have things called purines. And a purine is going to be your A and your G. They're going to have 2 rings. Again, this is going to come more in handy later. Your pyrimidines are going to have only 1 ring. That belongs in RNA only. So that's good to know. And when you talk about DNA or nucleic acids, let's see what we got. DNA is double-stranded. It's found in the nucleus. It's got the genes that are going to code for the proteins, all once again at the ribosome. The 2 strands are held together by hydrogen bonds, of course. I mean, really, right? We love talking about hydrogen bonds. And they run anti-parallel. The nucleotides have a backbone, which is going to be your phosphate group, sugar, phosphate, sugar, phosphate, sugar. And it is the bases that come off the sugar. So these are your bases. And A always goes to T, and G always goes to C. So that may be something to think about. A to T is going to have 2 bonds. G to C is going to have 3 bonds. That's why you can't simply mix and match one purine with pyrimidine. It just doesn't work that way. Nucleic acids, RNA is single-stranded and can leave the nucleus, and that's what's going to go on out to the ribosomes and eventually form, or well, begin to build a protein. Keeping in mind that RNA has uracil, so if they're talking about DNA and you see uracil, that answer is wrong. The sugar here is going to be a ribose sugar, not a deoxyribose sugar, so that's another breakdown. There you can see that this is RNA because it is single-stranded, and there is no T. There are U's. As for macromolecules, that'll do it. Now we move our story to water. Water, as we've gone on and on and on about, is made up of a polar covalent bond. So if that's an H, and that's an H, there's your oxygen right there. That is the polar covalent bond, which is going to set this up to be a partial positive, partial positive, partial negative. That is your polar covalent bond. That is not your hydrogen bond. The hydrogen bond will come over here when this water molecule forms a bond with the adjacent water molecule. So for example, it's the partial negative of that oxygen with the partial positive of that hydrogen. That's your hydrogen bond. Now, because of that, water has a ton of properties that are all very, very, very useful in nature. Water, the reason why this happens is because oxygen is electronegative, so therefore it sort of kind of hogs the electrons. It doesn't steal them, like in an ionic bond, but it hogs them, which is going to set this up. So once again, that's your polar covalent bond. That's your polar covalent bond. If you're going to have a hydrogen bond, it's here, between the H of one and the O of another. That's the hydrogen bond, so be careful on that. Oh, there it is again, right there. So we've seen that in class, so nothing new there. Now, because of this, water can do a lot of really cool things. First thing it can do is something called cohesion. And that is where water molecules will stick to each other. That's what allows water molecules to be pulled. We'll talk about in a second. Adhesion is what allows water molecules to stick to other polar substances, like the inside of a tree, which again we'll talk about in a second. Together, they're going to do something called transpiration. And that's how water can get from the roots of a tree all the way up to the leaves. Once again, I'll draw a picture in a second. Surface tension is this film that forms on liquid water. Once again, hydrogen bonds. That allows animals like the water strider to either walk or float on its surfaces. So in nature, that's pretty important. Water has an extremely high specific heat, so if you live in a coastal climate, if you live coastal, like for example Los Angeles or here on Long Island, you're going to have a moderate temperature, which means that the seasons are not extreme. Now, yes, we have, winter and we have summer, but they're not extreme. The heat of vaporization is the energy needed to evaporate water, and your body is warm enough to do that. So in doing so, you release the higher energy water molecules, leaving behind the lower energy water molecules, and that's what causes you to cool off. And ice floats. So because it floats, that means that the organisms at the bottom of the lake, say that's your ice right there, the ice floats, so that keeps the bottom, so little fishies can swim around, assuming they're not frozen, dead. They can swim around without being frozen solid like Austin Powers. And it actually, believe it or not, kind of keeps the really cold out. So let's say it's, -20 degrees Fahrenheit out here. It's kind of like a little insulator, which I know sounds crazy. You know that this water is going to be about 32 F, which again is cold, but it's pretty warm compared to negative 20. And that all happens because of the regular pattern or the lattice structure that forms, increasing the volume, decreasing the density. And the last thing I'm going to do, I just want to kind of show you how the transpiration works. So let's say there's a tree right there, there's the roots right there, there's the leaves right there. So what happens is, let's use some blue, when it rains, water is going to seep into the roots. And you're going to learn that through water tension. I'm sorry, not water tension, water potential. That's your H2O. Now because of cohesion and adhesion, this is going to be allowed to work its way up the xylem, which is the tiny set of microtubules that's found, well, not microtubules, but small set of tubes that are found in plants, which will then allow it to go to the leaves, where evaporation will happen. That's your transpiration end, and all the while pulling the water up from the roots with cohesion and adhesion. And the fact that it evaporates off the top, it allows it to free up. So hopefully that little review will be good. And let me know what you think.